DNA structure and function. Let's look first at the work that defined DNA as both a genetic material and a transforming principle. We will begin with Friedrich Michener, who was a physician working in the 1860s. He was the first person to isolate phosphate-rich chemicals, now known as DNA, from white blood cells, also known as leukocytes. He collected them by gathering up old used bandages from local hospitals, which must have been more than pleasant. He named the chemicals nuclein because they were isolated from the nuclei of the cells that he collected. Frederick Griffin was another scientist who studied DNA and its transforming principle. His question that guided his experiments was, can the material in dead bacteria transform living bacterial cells? 2 strains of S. pneumoniae were used in Griffith's transformation experiments. The R strain was non-pathogenic and didn't affect the mice. The S strain was pathogenic and caused the mice to die. When Griffin injected a mouse with a heat-killed S strain and a live R strain, the mouse died. The S strain was recovered from the dead mouse heart cells. Thus, Griffith concluded that something had passed from the heat-killed S strain to the R strain, transforming R into S in the process. Another set of experiments that gave weight to the theory of DNA as a transforming principle was completed by 2 individuals, Hershey and Chase. In Hershey and Chase's experiments, bacteria were infected with a phage, or a virus that only infects bacteria, that had been radiolabeled with either S35, which labels protein, or P32, which labels DNA. Only P32 entered into the bacterial cells, indicating that DNA was the genetic material. Individual nucleotides, as we now know, contain nitrogenous bases, sugars, and phosphate groups. The nucleotides are combined to one another by covalent bonds known as phosphodiester bonds or phosphodiester linkages. There are 2 major groups of nitrogenous bases, purines and pyridamines. The purines have a double ring structure with a 6-membered ring that's fused to a 5-membered ring. Pyridamines are smaller. They just have a 6-membered ring. The carbon atoms of the 5-carbon sugar are numbered 1' through 5'. The phosphate residue is attached to the hydroxyl group of the 5' carbon on one sugar to the hydroxyl group of the 3' carbon on the next sugar. Therefore, the individual nucleotides are attached to each other by a 5' to 3' phosphodiester bond. In the 1950s, Francis Crick and James Watson worked together to determine the structure of DNA at Cambridge University in England. Other scientists like Linus Pauling and Maurice Wilkins were also actively exploring this field. Pauling had discovered the secondary structure of proteins using X-ray crystallography. In Wilkins' lab, researcher Rosalind Franklin was using X-ray diffraction methods to understand the structure of DNA. Watson and Crick were able to piece together the puzzle of the DNA molecule on the basis of Franklin's stolen data because Crick had also studied X-ray diffraction and was able to decipher her work. In 1962, James Watson, Francis Crick, and Maurice Wilkins were awarded the Nobel Prize in Medicine. Unfortunately, by then Franklin had died of cancer as a result of her work with X-rays and could not receive the prize as it's not awarded posthumously. DNA is made up of 2 strands that are twisted around each other to form a right-handed helix. DNA base pairing takes place between an A and a T and a C and a G, or an adenine, thymine, guanine, and cytosine. They are complementary base pairs. The base pairs are stabilized individually by hydrogen bonds. The 2 strands are anti-parallel in nature. That is, the 3' end of one strand faces the 5' end of the other. The sugar phosphate of the nucleotides form the backbone of the structure, whereas the nitrogenous bases are stacked on the inside. Each base pair is separated from the other base pair by a distance of 0.34 nanometers, while each turn of the helix is 3.4 nanometers in height. Therefore, 10 base pairs are present at each turn of the helix. The diameter of the helix itself is about 2 nanometers, and it's uniform throughout. Only pairing between a purine and a pyridine can explain the uniform diameter. The twisting of the two strands around each other results in the formation of uniformly spaced major and minor grooves. Frederick Sanger was one of the scientists that allowed us to read the DNA chain base by base. In Frederick Sanger's di-deoxy chain termination method, he di-labeled di-deoxynucleotides and generated DNA fragments that terminated at certain points. This method is known as the di-deoxy chain termination method for that reason. The sequencing method is based on the use of chain terminators, those ddNTPs. The ddNTPs differ from regular deoxynucleotides because they lack that free 3' OH group on their 5-carbon sugar. So if you add the ddNTP to the growing nucleotide strand, the strand can't be extended any further. By using a predetermined ratio of deoxyribonucleotides to di-deoxynucleotides, it's possible to generate DNA fragments of different sizes. The DNA is then separated by capillary electrophoresis on the basis of size. From the order of the fragments formed, the DNA sequence can then be read. The actual readout is shown on an electropherogram, and it's generated by a laser scanner. We can then read the chain base by base. Now on to the specifics about that gel electrophoresis. It's a technique used to separate DNA fragments based on different sizes. The gel itself is made from a chemical called agarose. There's an agarose powder, and it's added to a buffer and you heat it up. After you cool it just a little bit, the gel solution is poured into a casting tray to solidify. You scoop out little wells, and into these you can load DNA. The whole thing sits in a liquid solution, and an electric current is applied. DNA itself has a net negative charge, and it moves via that electric current from the negative electrode towards the positive one. The electric current is supplied for sufficient time to let the DNA separate according to size. The smaller fragments move the farthest, and the heavier ones stay closer to that initial well that we created. Once the DNA is separated, the gel is stained with a DNA-specific dye for viewing it. You've already seen that picture. This allowed us to read a DNA chain, although it did take a while to figure out an entire genome. When we study DNA, we tend to study prokaryotes due to their simplicity and inferred ease of study. One of the first topics tackled was the mode of replication of DNA. Watson and Crick actually suggested a mode of replication in their original paper about how DNA was structured. Here are 3 models of potential replication. These models suggest that the 2 strands of the double helix separate during replication, and each of the strands serves as a template of which the new complementary strand is created. What was not clear at the time was exactly how the replication took place. There were 3 models suggested: the conservative model, the semi-conservative model, and the dispersive model. We now know that the semi-conservative model was the correct one. This was first discovered by Nielsen and Stahl. They experimented with E. coli, first growing in a heavy nitrogen, nitrogen 15 solution, and then a lighter nitrogen, nitrogen 14 solution. DNA grown in nitrogen 15, the red band, is heavier than DNA grown in nitrogen 14. And when you let it separate out, the sediments of the heavier DNA will rest lower in a cesium chloride solution when you put it into a special type of centrifuge. When DNA grown in nitrogen 15 is switched to a medium containing nitrogen 14 after just one round of division, the DNA will actually sediment halfway between 15 and 14. This indicates that brand new DNA after just one replication contains 50% nitrogen 14. In the subsequent cell divisions, an increasing amount of DNA contains nitrogen 14 only. This data supports that conservative model of replication. Here is what replication looks like at an individual strand level after the strands are opened and prepared to be copied. Before you try to write down everything I'm going to tell you, know that it's written in the text of the next few slides. So listen first, study later. To begin replication, a replication fork is formed when helicase separates the DNA strands at the origin of replication. The DNA tends to become more highly coiled ahead of the replication fork. Topoisomerase is an enzyme that breaks and reforms DNA's phosphate backbone ahead of the replication fork, thereby relieving some of that pressure that results from the supercoiling. Single-stranded binding proteins then bind to the single strands of DNA to prevent the helix from reforming before the job is done. Primase synthesizes an RNA primer that allows DNA polymerase III to synthesize a new daughter DNA strand. On the leading strand, DNA synthesizes continuously, whereas on the lagging strand, DNA is synthesized in short fragments called Okazaki fragments. DNA polymerase I replaces all those little RNA primers that help us start with DNA. DNA ligase then seals the gaps between the Okazaki fragments that formed, joining the fragments into a single DNA molecule. These are all the enzymes that I've just mentioned to you and their specific functions. Pause the video for a moment and write all of these down. Knowing them flat out is going to make your life a whole lot easier. Here are the 10 basic steps of DNA replication, or what I just told you, written out in a more simplistic form. Step 1, DNA unwinds at the origin of replication. Step 2, helicase opens the DNA forming replication forks, and these are extended bidirectionally. Step 3, the single-stranded binding proteins coat the DNA around the replication fork to prevent it from rewinding. Topoisomerase binds the region ahead of the replication fork to prevent supercoiling, which can actually break your DNA. Next, primase synthesizes RNA primers complementary to the DNA strand, and then DNA polymerase starts adding nucleotides at the 3' OH end of the primer. Elongation of both the lagging strand and the leading strand continues, although a little bit differently. On that lagging strand, the primers can't attach because it's running backwards, so it can't find the 3' OH end. Therefore, extra primers have to be added. Elongation continues, and eventually those primers can be removed by exonuclease activity, and the gaps are filled in by DNA polymerase by adding those deoxynucleotides. The gap between the two DNA fragments from all of those primers that just got taken away is sealed by DNA ligase, which helps the formation of phosphodiester bonds. These are the basic steps. Let's look again at a deeper level. We're going to use E. coli as our model system. E. coli has 4.6 million base pairs and a single circular chromosome, so it's capable of being replicated roughly every 42 minutes. Replication proceeds around this single circular chromosome in both directions at the exact same time, so the cell is adding roughly 1,000 nucleotides per second. This process does require a lot of energy, and it actually comes from extra phosphates on the nucleotides. DNA polymerases are the enzymes that dictate this whole process. DNA polymerase I and II are primarily required for repair, while DNA polymerase III is the enzyme that actually does the DNA synthesis. The whole process starts with DNA helicase. Helicase is capable of recognizing really rich 245 base pair repeats of adenine and then thymine. The helicase unzips the gene. Replication forks are therefore formed, and a single binding protein holds everything open. Now, DNA polymerase can only add in the 5' to 3' direction because it needs a 3' OH group to physically grab onto. So the DNA polymerase adds phosphodiester bonds between the 3' OH of one nucleotide to the 5' phosphate of the other. RNA primase adds RNA nucleotides, and DNA polymerase can hold on to these to start adding DNA nucleotides. Again, this addition is happening at about 1,000 nucleotides per second. Now, let's finally hit that lagging strand versus leading strand caused by the anti-parallel configuration of DNA. The leading strand is extended by primer alone. Once the DNA polymerase gets a hold of it, it can run through all the way to the end because it's never going to be disrupted. It always has a free OH to grab onto. However, the lagging strand has to start with a new primer every time the DNA polymerase essentially bumps up to the mechanism as the DNA itself is being opened. There's always a 5' end sticking out, so it has to build up into teeny tiny little pieces. There's a lot of activity happening as these different strands are put together. Here's what that looks like. This is what I mean. So you're uncoiling your DNA, and one strand is uncoiling perfectly. Your DNA polymerase can attach right on and read right down it. However, because your DNA polymerase has to move in the wrong direction back towards your parent DNA, you end up bumping up against the molecule. All of your enzymes have to let go, so you just create a whole bunch of little enzymes as you work on the little bit that you can, and then you have to wait, and you work on a little bit that you can, and you have to wait again. So there's a lot of activity. There's a sliding clamp that helps to hold the DNA polymerase in place, and the topoisomerase is preventing overwinding of your DNA. And there's a lot going on at once. All of the primers that have been created in your Okazaki fragments are eventually removed by exonuclease activities. The gaps are filled with deoxyribonucleotides, and DNA ligase seals everything back together. DNA replication in eukaryotic organisms is more complex, but it follows the same general rules. And that's because there are about 6 billion nucleotides in the average human cell and over 100,000 origins of replication. So there are many, many more polymerases to help get this job done. Here are some of the differences between prokaryotic and eukaryotic replication. As you can see, there are essentially many more enzymes, and the rate moves at a much slower pace because the eukaryotic DNA is essentially more valuable and has more potential errors. Our cells move slowly in order to assure there are fewer errors. Another big snag in eukaryotic replication are the telomeres. Eukaryotic chromosomes are linear, and the caps on the end of them are referred to as telomeres. A telomere is actually just made up of a TTAGG that's repeated constantly. We're talking like 100 to 1,000 times. These little telomeric caps can disappear, and you don't end up damaging your coding genes. It's kind of like adding extra clear coat to a car. You can rub some of that away without really damaging your paint. Same thing with all these extra nucleotides. But remember now, when you're replicating your eukaryotic chromosomes, you can only work in the 5' to 3' direction. So eventually, the replication fork is going to reach the end of the chromosome, and you've been building your Okazaki fragments piece by piece. Well, there isn't any place to add the primer anymore. You've kind of ran out. So those ends actually remain unpaired. In germ cells and adult stem cells, there's a special enzyme known as telomerase, and it helps to seal off those chromosome ends so it doesn't matter that they remain unpaired. Let's look at that process and then talk about why it matters. So you have a telomerase, and it's associated with RNA that complements that little tiny 3' overhang at the end of every chromosome. The RNA template is used to synthesize just a simple complementary strand. The telomere shifts, and then the process is just repeated over and over again. So primase and DNA polymerase can synthesize the complementary strand, and you're totally okay as long as you have this enzyme in your cells. However, we don't always want that enzyme in our cells. When our telomeres get short enough, that's a sign for apoptosis. That's telling your cell essentially, hey, you've had a lot of opportunity to divide, which means you've had a lot of opportunities to make potential errors. Maybe it's time for you to undergo apoptosis, and a newer, younger cell can take over, and we won't have to worry about those genetic mistakes. So telomeres shorten in somatic cells because they don't make telomerase as kind of a self-check. Shortening is associated with aging, and while the beauty industry might tell you that's a bad thing, aging is normal. Scientists have been messing with adding telomerase into the somatic cells of mice to see what happens. Just like in germ cells or stem cells, it extends the telomeres and reduces DNA damage. The cool thing is that it actually seems to be reversing neurodegradation, and it improves organ function, which tells you that adding telomerase might be a way to treat certain diseases. What we have found, though, when you alter the length of telomeres, you might cause cancer, because cancers have commonly been linked to an altered telomere state. Just be aware on this point that the book is wrong. The book is going to tell you that all telomeres or all cancers have shortened telomeres. Current research, current as in came out in August of 2016, however, tells us this isn't the case. Certain cancers, specifically lung cancers, have incredibly long telomeres, so don't get caught up by that as you prepare to do your homework. We have all of these opportunities for replicating our DNA, and we have all these opportunities for messing with our telomeres. What happens when it goes wrong? DNA replication is a highly accurate process, but mistakes can occasionally occur. Uncorrected mistakes may sometimes lead to very serious consequences, like cancers. Repair mechanisms are available to correct these mistakes. In rare cases, though, when they're not corrected, it can lead to mutations. In some cases, those mutations can even lead to errors in repair enzymes, so they themselves are mutated and defected. These cells are the ones that tend to become cancerous later on. Most mistakes made during DNA replication are promptly corrected. DNA polymerase immediately proofreads the base that's just been added. That way a correction can be made. The polymerase checks whether the newly added base has been paired correctly with the base pair on the template strand. If it's right, it just moves on. If it's not right, though, there's an extra enzyme that makes a cut at that phosphodiester bond, and the wrong nucleotide is just released back into the cell. This is all performed by the exonuclease activity of DNA polymerase III. Once that incorrect nucleotide has been removed, a new one is just added again, and the cell moves on. In mismatch repair, an incorrectly added base can actually be detected after replication has completely finished. The mismatch repair proteins detect this base and remove it from a newly synthesized strand by nuclease action. The gap is then filled back in with the correctly repaired base, and the cell can move on past the G2 checkpoint. Nuclease excision repairs thymine dimers. When cells are exposed to UV light, thymines lying adjacent to each other can actually form thymine dimers. They react with each other rather than a base across from them. In normal cells, they're regularly excised and replaced no problem. A well-studied example of mistakes not being corrected are seen in people studying from ectoderma pigmentosa. Affected individuals have skin that's highly sensitive to UV rays from the sun. When individuals are exposed to UV, pyridamine dimers, especially those of thymine, are formed. People with ectoderma pigmentosa are then unable to repair this damage. They're not repaired because of a defect in those nucleotide excision repair enzymes, whereas in normal individuals, as I mentioned, the dimers are just excised and the defect is corrected. The thymine dimers distort the structure of their DNA double helix, and this may cause problems during DNA replication. People with ectoderma pigmentosa have a much higher risk of contracting skin cancer than those who don't have the condition. Errors in DNA replication are not the only reason that mutations arise. Mutations, or just variations in the nucleotide sequence of a genome, can occur because of damage to the DNA itself. Induced mutations are those that result from exposure to chemicals, UV rays, X-rays, or some other environmental agent like asbestos. Spontaneous mutations occur without any exposure to an environmental agent. They are just a result of the natural reactions that take place within our bodies. Here are 3 examples of simple mutations. There are point mutations that are silent, so they have no effect on the protein sequence. There are missense mutations, and they result in a single amino acid substitution. They might not seem very big, but cystic fibrosis is caused by a missense mutation. In nonsense mutations, a stop codon is substituted for an amino acid. That means that any potential protein made is going to be far larger than it ever should have been, and it's going to greatly affect its function. A final mutation to mention in this lecture are frame shift mutations. Insertion or deletions of nucleotides may result in a shift in the reading frame or an insertion of a stop codon. This is kind of like if you had a big paper or a sentence that you were typing out and you changed where the spaces were in the word and you didn't have any options to change it back. This can greatly alter the way a sentence is read, and this can greatly alter the way a protein is read. This essentially destroys a protein. While it may give it some new, novel, crazy function, it's most definitely not going to look as it should before. I realize that this chapter is extremely concept heavy, so do me a favor, go back and read it and try to make an outline. Take your own notes and then attempt to do your homework assignment, working through the problems individually. Don't forget, within Learner on the bottom right-hand corner, if you click the Resource button, it takes you directly to the area of the text where you will find the answer. Clicking on the Resource button does not decrease your grade on the homework assignment. It's there to help you, not to hurt.